The film Gattaca was released in 1997. It starred Uma Thurman, Ethan Hawke, and Jude Law. And this movie portrayed a dystopic future where an individual's identity was validated or invalidated daily by high-resolution, rapid DNA sequencing. When I was a graduate student, I thought this technology was absolutely preposterous and truly relegated to the realm of science fiction. Let me explain to you why. The human genome contains 3 billion nucleotides, A, C, G, and T, and you can think of these as the genetic building blocks of our humanity. And they're strung together in sequences like this along our 23 chromosomes. And amidst these 23 chromosomes are 20,000 genes. This image here that I'm showing you is called a karyotype. It's the DNA content of a single cell, and you can see that it's missing the Y chromosome, so it's from a woman. But the interesting thing about this sequence is the complexity that it presents. Particularly when I was a student in 1997, we could barely sequence 800 nucleotides reliably in the lab, and this would sometimes take 2 to 3 days. That's a far cry from 3 billion nucleotides that make up the human genome. And in keeping with the difficulty of sequencing, it took nearly $3 billion and more than 200 scientists to release the first draft of the human genome in 2001. And you can see that over time, the price of DNA sequencing declined very slowly. This follows Moore's law. But then there was a cliff, and then these sequencing technologies improved very dramatically and became very, very inexpensive, such that this year there's been much publicity about sequencing an entire human genome for $1,000. And I bet you by the time that the Christmas sales roll along, it'll be cheaper than that. So I would like to share with you today 2 short stories from my lab about how this affordable, high-resolution genome sequencing is rapidly changing the face of biomedical research. And my favorite organ in my lab that we study is the pancreas, and it's very appropriate that we discuss the pancreas just after lunch. Because after you've eaten, your body is metabolizing those carbohydrates and yummy Indian food into sugar, and this is going to trigger your pancreas to release insulin, and insulin mobilizes sugar uptake into your muscle and your fat. And the two approaches in my lab to study pancreatic function are 1, an embryological approach, and 2, a physiological approach. So my lab for many years, since I've been in Singapore, now a decade, has been interested in the genes that are activated during embryonic development that lead to the construction of this indispensable organ that we all need for life. But in humans, there are very, very rare mutations that lead to a condition called pancreatic agenesis. And agenesis means failure to form or failure to form properly. And I wanted to give you an idea about what this looks like by examining this disease in another organism, one of our models in the lab, which is the mouse. So here you have 2 mouse pups around 1 day old. You can pretend that they're sleeping. And the pup on the top is a normal pup. Everything is physiologically normal with this animal. The mutant on the bottom, we've inactivated a particular gene that is responsible for the construction of the pancreas. And without this gene, the animal doesn't have an organ and cannot produce insulin. But you can see this little pup was able to suckle with its mother, and it has milk in its stomach. So behaviorally, it's normal. But a few days later, what happens is this little guy or little girl goes through a tremendous wasting syndrome, and the reason for that is without insulin, the blood sugar levels of this animal are through the roof, and this is incompatible with life, so the animal will die. And this is exactly what will happen in the rare human patients that have this disease called pancreatic agenesis. Given that we have had a long-standing interest in the genes that are responsible for organ building, for building the pancreas, we were contacted several years ago through a collaborator in the Middle East where a physician had encountered a family where a young baby girl was born who was apparently normal at birth, but within several hours began to show neonatal diabetes. And the physician was really intrigued by why this young girl was suffering this condition so soon after birth. They did x-rays and they found that the child did not have a pancreas. But the physician wanted to know why. What is the genetic lesion that leads to such a catastrophic clinical presentation for this young baby girl? So very much like CSI and all those other programs that you watch on television, you can take a saliva sample, extract the baby's genomic DNA, and through genome sequencing technologies, we were able to pinpoint the mutation in this young baby girl's DNA that led to this very dramatic loss of an organ. And we were able to find that there was a single nucleotide change in a gene called RFX6, is a gene on chromosome 6, and there was one single nucleotide change in this young girl's DNA. So to put this in context for you, remember I told you that the human genome is 3 billion nucleotides, and just one change in this young girl's DNA led to this catastrophic loss of an organ and invariably led to this young girl's death. And this is like the proverbial needle in a haystack, but these genome sequencing technologies that are now affordable and high resolution allowed us to have rapid access to the genetic lesion for this clinical presentation that happened in this hospital in the Middle East. And we found this to be quite remarkable, that it was indeed just a single change in the 3 billion nucleotides. This information also allowed us to inform the parents that they're indeed carriers for a very rare mutation that leads to this disease called pancreatic agenesis. And so when the parents became pregnant again, the question became, what is happening with their unborn child? And with the information that we had provided them that was developed in Singapore, they were then able to undergo genetic testing. And this is by taking a small biopsy from the unborn child and then doing prenatal genetic testing in the Middle East to find whether or not this unborn child is actually carrying the mutation. And regrettably for this family, that was the case. And so the information that we were able to identify in Singapore empowered this family to make a choice, whether they wanted to continue with the pregnancy or whether they wanted to save themselves the emotional distress of having a child that would die soon after birth. And I'm not telling you this rather grim story to depress you and to sadden you. I'm just trying to explain how rapidly a clinical presentation in the Middle East can lead to a diagnosis in Singapore, and you hone in on a gene that had been previously uncharacterized for this, had previously not been associated with this dramatic clinical presentation of no pancreas. But there are many genes, single genes, in which mutations lead to many different diseases. And in fact, they're cataloged weekly at this website called the Online Mendelian Inheritance of Man. And I captured this data from the website just this week, and you can see that there are roughly 3,300 genes that have been shown that when mutated lead to a presentation of a particular disease. And remember, I showed you the estimate in my first slide. There are roughly 20,000 genes in the human genome. This is roughly 1/6 of our genes have been shown that when mutated give rise to a disease. So what are these? You're familiar, I'm sure, with cystic fibrosis, sickle cell anemia, Huntington's disease, hemophilia that affected 19th and 20th century European royalty, albinism, dwarfism, and for those of you that were maybe a bit older, you might remember a film about the boy in the bubble who had an immune disorder, which was also due to a mutation in a single gene. So with our ability to screen for all of these different mutations, this raises a number of ethical questions that I cannot answer for you today, but as young people and parents, and as you become parents and grandparents, you are going to be confronted with as the accessibility of genome sequencing becomes even more widespread and even more affordable. So for example, this image on the left-hand side of the screen shows a human embryo, which is at the 8-cell stage. It's immobilized with a little suction pipette. And if you have really good video game skills, you can go in with a microinjection pipette and suck out one single cell from this human embryo. And using these sequencing technologies that I've described to you, you can then assay those 3,300 genes for mutations. So you can screen before transfer if you were performing IVF for the embryos that are quote unquote desirable. This raises a number of really challenging ethical questions because what do you consider to be disabled? For example, the ethical question of the ethical situation of when thinking about an embryo that might be blind or deaf presents to challenges that many blind and deaf individuals lead rich, long, colorful lives. So how could you possibly eliminate an embryo that might be blind? And so again, these are questions that I cannot answer for you, but as this technology that I'm describing to you becomes more pervasive in our lives, you may in your own family be forced to confront these kinds of technologies that I use in my laboratory, but might affect you as individuals. And so I'm going to give you a last short story about high-resolution genome sequencing, again returning to the pancreas, and this to the disease that you all know about, because more than likely it affects your family members as well as mine, and that is diabetes. Diabetes is a global health burden. It's estimated that 370 million people worldwide have diabetes, and it's really quite hard to get your head around exactly how many people that is. I mean, it's just almost inconceivable. And in Singapore, where there are really, really good medical records, it's been shown that there are roughly 11% of Singaporeans between the ages of 18 and 16 have diabetes. In the United States, the estimate is roughly 10% of the population, or 29 million, have diabetes. And to put this into dollars and cents, in 2012, it was estimated that it cost $262 billion to manage diabetes in the United States, and that far exceeds the GDP of Singapore. So it's truly a burden to treat and sustain people who are diabetic. There are two flavors of diabetes. You're more familiar with what is called type 2 diabetes, which is oftentimes called lifestyle or obesity-related diabetes. And in this scenario, clinically, the pancreas either produces too little insulin, or your body suffers from insulin resistance, where those tissues that should respond to the insulin signal don't seem to respond appropriately. My laboratory has been focused principally on the type 1 diabetes, which is an autoimmune attack where, for unknown reasons, your body turns on its pancreas and kills off those essential cells in your pancreas that produce insulin. And without that, those cells that produce insulin, you become insulin dependent, and you have to have insulin jabs for the rest of your life. We know from studies globally that there is a genetic component to this disease, but the reasons that type 1 diabetic patients develop the disease in the first place has really been a mystery. And given our collaborators in the Middle East, we were introduced to a family where there was a high proportion of type 1 diabetes in their children. And this family, I'm showing you a typical pedigree that we would show in lab meeting. Squares are men, circles are women. So this is mom and dad. There's 7 children. These are the ages of the children roughly 2 years ago. And the solid squares and circles represent the children that have type 1 diabetes. And so why this family was significant is this family is from a closed community. It's not like the multiracial discussions we were having earlier. These are mothers and fathers that marry within a very tight community. Therefore, they are genetically related, and their children will be genetically related. And this means for someone like me, who is a geneticist and has access to genome sequencing, we can ask a simple question: well, what distinguishes this unaffected child from the affected child? And can we use our technological platform to begin to decode which genes are responsible for this very devastating disease of type 1 diabetes? So what do? We genome sequenced. And in this case, I can tell you this was done in the last several months. It really did cost $1,000 to decode the entire 3 billion nucleotides of mom and dad and a couple of the children. And through these studies, and using a lot of help from the computer guys, we have honed in on a region of chromosome 7 that segregates with the disease. This means that the affected children have a particular region in common that the unaffected children do not. And so what is that region? Well, it's not as simple as the story I told you about pancreatic agenesis, where it's one change of a nucleotide over 3 billion. We have 6 million nucleotides to begin to study. And within that 6 million nucleotides, there are 50 genes. So my lab and I, we have our work cut out for us. And so we are beginning now high-resolution studies of this region of chromosome 7 in an effort to identify the gene that is responsible for this very devastating pathology of type 1 diabetes. And it's our hope that in understanding the genetics of this disease, this will open doorways to the development of drugs and other therapies that might prevent the disease in the first place. And with that, I can think of no better motivation for me and my team to go back to the lab and start all over again on Monday morning. So I would just like to thank you for your attendance, and my teammates and collaborators, and most importantly to the families that participated in this research. Thank you.